Okay. Yeah, it's a pleasure to finally present this work here in this context. It's basically our contribution to the special issue in which we showed that silencing hippocampal region CA2 actually affects spatial learning. And yeah, let me just provide the background. So we heard several times, and probably all of you know, that CA2 is essential for social recognition memory. But what about non-social memory? And there are a couple of reasons why we think it should be involved initially. And one of the reasons is that if you look at the projections and the subpart of the hippocampus involved in social recognition memory, you can see that the CA2 is dorsally, of course, involved in social recognition memory, but it projects dorsal CA1 and dorsal CA3, which have been shown by work from McHugh and others, that they are not important for social recognition memory. But if you basically look in literature, what they are very prominently involved in is spatial learning. So that was the first hint in a way. And then if you look at other articles and then work, for example, by George Alexander, you see that if you place an object somewhere, that CA2 spatial representations strongly remap. So it could be that they're involved in forming spatial representations of also spatial locations. And then there's work by the McHugh lab again, Böhring et al. 2017, where they show that contextual habituation is slower in mice with CA2 without CA2. And that means basically that you put an animal into some kind of enclosure, and then if you repeat that and it becomes familiar, that basically the subject animal is not interested as much anymore to explore. And it's not that CA2 silencing completely abolishes it, but it somehow seems to slow this process down. So maybe CA2 is required for some kind of efficient learning. And then there's work we just heard that Hongshen presented and has also published before. When you silence CA2 transiently, that you see basically that reactivation quality during sharp wave ripples is impaired. So of two linear tracks. And given the importance of sharp wave ripple reactivation for spatial learning, we would think that, okay, that should have an effect on spatial learning. And then there's another interesting insight from Hongshen, and is that if you silence CA2, that during individual sharp wave ripples and also outside ripples, there's more co-reactivation of two distinct experiences. So those two linear tracks, which have been different linear tracks, they are reactivated in a single sharp wave ripple event. And so it may be that CA2 may help to separate different memories. And if they are somehow co-reactivated, it may be difficult for animals to somehow distinguish between different memories. Okay, so we thought the ideal data for that would be actually the Wars motor maze, since it's some of the gold standard for hippocampus-dependent spatial learning. And for those of you who have not heard about it before, it's basically you put an animal into some kind of water enclosure, and there is a hidden platform which the animal is not able to see or smell or so on, and it somehow has to learn over time to locate this platform. And the more you put the animal inside this, the more trials you have, the better the performance gets. And you would think that, or we would think that different phases of the spatial learning paradigm require different abilities. So in general, you basically have some experience, and then based on this experience, you build an internal representation that allows that kind of navigation, that you know where the platform is. And upon learning, so the normal phase is the following. You have some cued phase where basically you have a flag that shows the animal, okay, there is a platform. So it learns that there is a platform and it learns to swim there. But then you have a learning phase where you move the platform location. And during this learning phase, somehow the animal has to detect there's something novel happened. There is actually a platform without this flag. And then it has to form the spatial representation. And then you often have a probe trial just to see whether the animal keeps swimming there without the platform. And then after a probe trial, normally you have a reversal learning phase where you change platform location. And during the reversal learning, that basically novelty has to be detected again. Somehow the animal has to learn that the old platform location is no longer valid, and you have to form a new spatial representation. And to do so, you probably have to distinguish between what is old and what is new. So this distinction comes in place again. So we formed very clear hypothesis, we would expect that CA2 silenced mice should actually be somehow impaired in navigating to this hidden platform, both in learning and in the reversal learning, and that this impairment should be particularly strong during reversal learning, since then we have two distinct experiences. But there's a but here, and that is that in this original paper by Hitti and Siegelbaum 2014, they actually tested spatial learning in the Boris motor maze, and they found that there's no significant differences looking at the path length, so how long the animal took on average to find the platform, or on latency. And that somehow, yeah, really, it somehow went against our intuition. So we asked them that they can share the data so that we can dig deeper in, because latency and path lengths are somehow the basic ways to analyze this data, but there are more elaborate ways to do so. So we thought, okay, let's look more closely at that, and maybe we find something. And yeah, actually, we first plotted the occupancy maps, and so basically just the average occupancy of an animal, of basically averaged across all animals across certain days, so averaged across trials and so on. And during learning, we didn't really find the difference just visually. But when you look at reversal learning, we started to see a visual difference, so especially in these first 2 days. So the CA2 silenced mice spent much more time around the old platform location here by this dotted line compared to the control line, control mice. And that also you can still see on the second day. So we quantified that. And so during learning, we looked at different measures than before. We didn't look at the distance swam or time to reach the platform, but for example, to distance to the original platform on average. And during learning, you basically see that the original platform, the animals just get quicker and quicker swimming there, but there's no difference between CA2 silenced mice or control mice. However, during reversal learning, we see like a consistent difference. So basically, the more they learn to swim to the new platform location, they should increase the average distance to the original platform. And CA2 silenced mice seem to be consistently somehow attracted to the old platform location. So you see this somehow, yeah, distance doesn't increase as much. And correspondingly, if you look, for example, the time the animals swim in the target quadrant, like basically changing from the original to the new platform location, you also see a systematic difference there. So the animals, basically, CA2 silenced mice spend less time in the new target quadrant compared to the old one. Okay, so that somehow basically was a reason for us. Okay, we see differences there, and now let's look a bit more in detail what differences there actually are. And there's a way to somehow classify the behavior of the animal. What kind of strategy do they use? And there is a software package online which has been trained by other people to somehow basically, given a certain trajectory of animal behavior, what kind of navigational strategy that is. And so we didn't set any parameters here. We just took this algorithm, fed in our trajectories, and it said what is the most probable strategy here. And it uses some kind of random forest classifier. So for us, it was nice because then we didn't have to manually decide on any parameters here. And normally, you can say basically when the animal starts learning, it uses non-spatial strategies. And if it gets better and better, the more it uses spatial strategies. And initially, it starts with something like called thigmotaxis, which is swimming, circling at the border somehow. And then it somehow starts scanning more efficiently. And when it uses spatial strategies, it somehow keeps swimming more centrally to the target and then searches there. And the ideal case is that it swims directly to the target, which is called direct path. However, during reversal learning, you see also that kind of perseverance behavior, meaning that it swims to the old platform location and somehow keeps swimming there, even though it should have realized that the platform is no longer there. Okay, when we apply that to the data, we somehow can confirm what we saw before, that we don't see a difference during the initial learning. So there was no significant differences between the two mice, neither for any of the strategies we looked at. However, during reversal learning, so this is basically now after platform has been moved, we see differences. So we see, as you can visually always see, the perseverance behavior is much stronger in CA2 silenced mice compared to control. It's not a super strong effect, but at least it reached levels of significance. And what you can also see, if you look at that, the spatial, the non-spatial behaviors, which are here more in blue versus the greenish spatial behaviors, that they are much more prominent on the second day of CA2 silenced mice. So that also confirmed that, especially in the initial phase of reversal learning, CA2 silenced mice are impaired. So our conclusion is that CA2 silenced mice, or CA2 is reported when the animals flexibly adapt the behavior. And yeah, we find that CA2 silenced mice spend more time near to the all platform location, show increased perseverance, and so somehow stick longer to non-spatial search strategies. However, we could not, or we had to say, okay, our initial hypothesis that this effect should also be there during initial learning, that we didn't find any evidence for that. It could be that it's chronic silencing, or it could be partially silenced, we don't know. So yeah, there are different explanations for why that actually may be the case, like on a circuit level. So there is this proposal by Middleton and McHugh that CA2 and CA3 could be additional input structures. So you have very prominent and strong cortical input to CA2, as we heard before. And that could be somehow like a second way of how information flows to CA2 or to the hippocampus. And there is a strong mutual inhibition with CA3. And yeah, Middleton and McHugh coined this term like memory versus sensory-based routing. It could be that while CA3 somehow maintains memories, that's direct projections from entorhinal cortex to CA2 could somehow facilitate new sensory information. Then if something is detected as novel, that somehow CA2 suppresses CA3 and then sends this projection, this information directly to CA1. However, there could be an alternative explanation, and that is what we have been putting forward, and that is that CA2 and CA3 may act actually as a functional unit, so that both this mutual suppression, but also positive interactions could play a role. And the idea is that you have cell assemblies in CA3 and CA2, which can pair, and somehow this pairing allows for prioritization later on. And then there's also this inhibition aspect that if you have CA3 and CA2 paired, they can inhibit better other memory traces. There may be a totally different explanation there as well, because there is actually data that newborn cells from newborn cranial cells may also be involved in this increased perseverance behavior. So we know that newborn cranial cells project to CA2. If you suppress newborn cranial cells, you also see this increased perseverance. And CA2 cells project to ventral hippocampus, which has been shown if you lesion ventral hippocampus, you also see this effect with delayed onset on spatial learning strategy. So it could be also a totally different explanation. We don't know by now. However, if you want to experimentally test it, one could basically distinguish between our approach and the idea from Tom and Middleton is that, so if the sensory versus memory-based routing is true, then you would imagine that activity in CA2 should somehow carry information about the old platform location, while CA2 should carry information about the new platform location. And that with novelty, activity of CA2 should increase and CA3 activity should decrease on average. If it would be CA3-based prioritization, we would imagine that CA3 and CA2 cells somehow like have coordinated reactivation after such an experience. And that neuromodulation and plasticity, as are the key ingredients of our proposal, that they are somehow required for this process. Okay, cool. With that, I want to thank you for your attention, and I want to say that this has been joint work with Andrew Lehr, Fred Hitty, and Scott Dybel, and we're very grateful for Fred Hitty to share his data and let us take a new view on that. Thanks.